Hello everyone, welcome to part 2 of chapter 3. Now that we have explored some of the laws of thermodynamics, including the first one that states that you can neither create or destroy energy, you simply transition it from one state to the next, and the total amount of energy is constant in a closed system, as well as the second law that says that the transfer of energy is not 100% efficient because you tend to lose a lot of it in the form of heat. Let's go ahead and take a look at how we can go ahead and apply these laws into things like chemical reactions. And what does it mean when we talk about things like free energy, delta G, equilibrium constant, as well as the role that enzymes play in all this as a biocatalyst. All right, so first things first, it turns out that if you take a look at chemical reactions and you solely focus on their energy utilization, we see that they tend to proceed in the direction that will cause a loss of free energy. Now, what exactly is free energy? Well, by definition, free energy is going to be energy that you can harvest to do work or drive chemical reactions. And oftentimes, free energy is generated by the fact that we have to break chemical bonds, like for instance, going from a protein to an amino acid. And as you break those peptide, covalent, ionic bonds, what you're doing is you're releasing the energy that's being harbored by the electrons that were holding the molecules together. And it turns out that for many, if not all, chemical reactions, they will proceed in a direction that will lead them to a loss of free energy. So they're going to be releasing that free energy because that is what we label an energy favorable reaction. Now, an energy favorable reaction would be, for instance, and you know what, I'm going to steal the example that the book gave. It basically says, let's go ahead and assume we have a sheet of paper and we're going to apply a lighted match to it. Well, the combination of the lighted match, paper, and of course oxygen in the air is going to cause the paper to start burning and generating smoke. And along the way with the smoke and the ashes, it also releases heat and carbon dioxide. Now, the products of this reaction, so the fact that I start off with my paper, my match, and my oxygen molecules, and I am able to produce smoke, ashes, heat, carbon dioxide, even some H2O. All of this is an example of a release of free energy. I'm releasing energy that can potentially be harvested to do work and drive other chemical reactions. And that's not even heat, that's also including heat because heat's not really lost. It's just a form of irretrievable energy that's dispersed that we can't really utilize. Now, because it is easier for the paper as it burns to be broken down into all these components, that flow of the reaction, the release of the free energy, that would be an example of an energy favorable procedure or an energy favorable reaction. It is very unlikely that the smoke, the ashes, the heat, the carbon dioxide molecules are all going to spontaneously combine together and regenerate that sheet of paper. They won't be able to do this because in order to do that, you're going to have to require a massive amount of energy to put back into the system to reverse that initial reaction. So the fact that the smoke, the ashes, the heat, the carbon dioxide can't go backwards, can't reverse, that would be an example of an energy unfavorable procedure or reaction. Now, in order to assist us with this loss of energy, use of energy, what we see happening is that a lot of times molecules will rely on enzymes to reduce the energy that's needed for these spontaneous reactions to occur. And the thought process about this is that most reactions have what we call an activation energy. And an activation energy is basically an energy barrier that needs to be compensated for the reaction to occur spontaneously. Usually this might require an input of energy that the cell simply is not able to generate at a spontaneous level. And when I say spontaneous, it goes back to the fact that all molecules have kinetic energy, meaning that they are always in this constant state of motion. And what we want to do when we have a chemical reaction is we want to have the molecules generate so much movement that they're able to generate their products, whether that product is for the molecule to combine and build something larger, or for that molecule in essence to split and go from a polymer to a monomer. So regardless of what you're doing, whether you're doing a dehydration synthesis where you are building, or you're doing a hydrolysis reaction where you're pulling it apart and you're making something smaller, you will always have an activation energy that you require for you to kind of push through and get that reaction to start. In my little paper example, my activation energy was provided by the heat of lighting the match. So you needed that energy from the heat to go ahead and get the paper to change into an alternate state, which obviously would be the ashes and the smoke. Now, in our body, obviously, we're not going to light a match. So what we see happening is that even though temperature does have an effect in increasing the movement of the molecules, if you increase your body temperature, molecules tend to move at a faster rate. We really can't hike our temperature up that much without going into our fever category, which of course could be detrimental to the whole system. So we can't use temperature, but what we can use are the enzymes. And the way the enzymes are going to work is that they're going to lower our activation energy, and by lowering the activation energy, they're going to increase the likelihood that the substrate will go ahead and collide and be able to produce the product that we are after. So let's go ahead and take a quick look at the illustration that is on our slide right here. And as you can see, we have our reactant, which is our starting point. That's Y, and we're trying to get to X, which is our product. Now, it turns out that in order for us to do this, we're going to need to get over this little barrier indicated right here. The difference is between A and B, and this is our activation energy. Once we're able to generate the activation energy, that means that there's enough random movement within the reactant to go ahead and produce the product. Now, look at the difference in activation energy required, or the barrier, between what we call an uncatalyzed and an enzyme catalyzed pathway. So in our second observation in B, we're going to utilize enzymes. And as you can see, the barrier has been severely reduced. Thereby, the activation energy has been significantly lowered, making it much more likely for Y to generate enough energy, enough activation energy, and the synthesis or the reaction to occur so that you can get to your final product, which is labeled X in this case. Here is another example where it just talks about that if we're lowering the activation energy, the whole purpose of that is that you want to increase the probability that a reaction will occur. So you can see that if we have an enzyme catalyzed driven chemical reaction, the energy is severely reduced. Thereby, it makes it more likely that at room temperature, or even at our internal temperature of 37 degrees Celsius, you'll have a much more likelihood that you'll have enough kinetic energy within the substrate that it will be able to surpass the activation energy and thereby drive the reaction over to a favorable rate. Obviously, in this case, you want to go ahead and you want to keep in mind that your catalyst is increasing your rate, but we're not actually utilizing the we're not using up the catalyst. We'll be able to recycle that enzyme over and over again. We also do not alter the product that is generated in the reaction. The product is exactly the same with or without the enzyme. The only thing that you're altering is the rate of the reactions. Some enzymes have the ability to multiply or increase the reaction rate by trillions of, by speeding it up a trillion times. So it's a huge difference, and you know, we did cover chapter 4 previously when we were talking about proteins. So I know you were very well versed on enzymes and the fact that they're able to go ahead and catalyze these reactions by lowering the activation energy. So just a quick reminder, remember that when you take a look at enzymes, the majority are protein-based, so they will have to follow certain rules. And when we look at the protein-based enzyme, we see that they will have an active site that will be specific for the substrate that they're able to interact with. The substrate, it's almost like a lock and key analogy. Remember, it has to fit properly, utilizing some non-covalent bonds. And as it fits, what we see happening is that we form an enzyme substrate complex. And at this point, when you have full interaction between the enzyme and the substrate, we see that the chemical reaction starts to occur. So you can see they've labeled it as the catalyst. is working on it. So we're lowering our activation energy and we're driving the reaction to go from substrate to product. And once that is done and the enzyme product complex has formed, we see that the bonds will start to loosen between those two. And the nice thing is that your product is released and your enzyme stays fully intact, so it gets ready to be recycled over and over again. Also, what you want to keep in mind is that with enzymes, it's all about optimal conditions. So there really is some such thing as like an optimal temperature, meaning a temperature point at which it's able to produce its maximum amount of products most efficiently. And usually what we see is that if we slightly increase the temperature, the enzyme will go ahead and work faster, generating more products in a shorter time period. However, you have to be very careful with an increase in temperature, because if you make it too hot, you will damage the non-covalent bonds that are interacting with our peptide bonds, and therefore the enzyme can denature, meaning it will lose its shape and thereby also lose its structure. And for those of us wondering what happens if I lower the temperature, well, we see a reverse in action. By lowering the temperature away from the optimal setting, we see that the enzyme will slowly start to slow down, and eventually it will shut off if it's too cold. However, it's very easy to regenerate that enzyme by simply spiking up the temperature and allowing it to once again form its interaction with its substrate. Obviously, besides temperature, there can also be adjustments to the pH, the salt content, anything that will really alter or change the optimal environment the enzyme can affect its production rate. Now, another term that you're going to come across as you're perusing through the chapter is what we like to call free energy. Free energy is often indicated by just a G, and free energy is basically useful energy in a system. So this, for instance, is going to take a look at energy that we can utilize to thrive and to continue to use in another process. So for instance, we can see that we can utilize free energy to figure out if we're taking a look at an energy favorable or an energy unfavorable reaction. So on your notes it says that the free energy change for a reaction will determine whether it can occur. Now, there's a difference between free energy and free energy change. Free energy change, let me just go ahead and highlight that for you. Free energy change is often indicated as delta G. There you go. Okay, and what we see happening is, now we can take a look at our illustration right here, is that you can have an energy favorable or unfavorable reaction. It will all depend on where the free energy is greater, within the substrate or within the product. So in my first example, I see that I have my Y and I have my X, and notice how I want to have my reaction proceed from Y to X. My apologies. I forgot my letters there for a second. So follow the arrow. You want to start with Y and go over to X. And if you come over here to the explanation, it says that the free energy of Y is greater than the free energy of X. Therefore, your free energy change, your delta G, is negative. And that means that the reaction can occur spontaneous, especially because as it's occurring, we see that we start increasing the amount of disorder in the universe. So that, by all definitions, is considered an energy favorable reaction. So when the reaction can occur spontaneously, and we see that there is a loss of free energy, right? Because you're downgrading from Y to X. So you're going to go from a high energy point, a high free energy point, to a low free energy point, thereby making your change of free energy, your delta G, negative, right? Because you're losing it. And as you're losing your free energy, you're generating more entropy, more disorder. This is what we like to see. Well, it's not what we like to see. This is the easiest way to do a reaction. So as we can see, it's going to be energy favorable. Now, what if you want to go the other way, right? So what if I want to go from X to Y? Notice the little red arrow. Well, in this case, we have a slight problem because as we can see from the written part, X is going to be at a lower energy state than Y is. So in order for it to transition, we're going to need to have a free energy change that would be positive. And if that happens, that will require the input of energy, and we see that there's a reversal in the amount of disorder, so we become more ordered, not disordered. So this would require an input of energy. Energy has to go in here. And because we have to put energy into the system for the reaction to occur to get to our final point, we see that this is an energy unfavorable reaction. Now look what it says over here. It says this reaction can occur only if it's driven by being coupled to a second energetically favorable reaction. So your body has figured out a way how to do an energy unfavorable reaction by oftentimes coupling it to an energy favorable reaction. Both of these reactions tend to go ahead and piggyback of each other, and then the energy generated in one will then be immediately used to continue the process for the secondary one. This brings us back to one of our previous chapters where we talked about the fact that you have your dehydration and you have your hydrolysis processes. You have your analysis and your catabolism process, meaning that as I am destroying my macromolecules, for instance, in my digestive system, I am basically going from macro to monomer. I am going to a lower energy level. I'm releasing the energy that's being held in the bonds for my macromolecules. As I'm doing this, this is an energy favorable reaction, and I can take that energy and I can put it immediately into a reaction that's going to require me to put energy into it, such as my dehydration or my condensation reaction, where I'm actively building, I'm forming new bonds. In order to do that, to form these new bonds, I'm going to have to put energy into the system. I'm going to have to put effort into getting it all done, making it an energy unfavorable reaction. So oftentimes what we see, and obviously there will always be some exceptions to the rules, but oftentimes what we see is whenever you're looking at a reaction where you are destroying, where you're going from large to small, it will have this burst of free energy that's being released, allowing your free energy change, your delta G, to become negative because you're going from high to low. And as you're doing this, you're causing an increase in entropy because remember, as you're transitioning from a higher energy state to a lower energy state, it's not 100% transfer. You're losing some of that energy in the form of heat, making it all energy favorable. And then what I can then do is I can turn around and take this release of energy and I can plug it into a system that will require energy to go from substrate to product. And whenever you're plugging in, you're in essence putting in extra effort, which means that oftentimes you are building. So we're going from amino acids to protein. We're going from glucose to carbohydrates, nucleic acids to nucleotides. As you're doing this, we're going to see that our change, our delta G, is positive. We're going to see that we have to go over to a higher energy point, and in order to do that, we have to label it as an energetically unfavorable reaction. So oftentimes what we do is we couple it. One will depend on the other. In fact, if you go over to your next slide, here we see a little reaction coupling, and notice how the one with the negative delta G is able to generate excess energy. So here's the excess energy that's being generated in my illustration, and I can use that to thrive or to fuel the energy unfavorable reaction. All right, so this is one of the ways that we go about. Remember, we don't lose, we don't destroy energy. We simply transfer it from one system to the next. So in this case, what we're doing is part of the product production of when you do a hydrolysis reaction or a destruction reaction or an energy favorable reaction. Part of that is that you generate lots of free energy, which we can then go ahead and plug into an energy unfavorable reaction, which will often constitute the fact that we're putting in effort and we tend to build and get a bit of a larger complex. In order for the coupling to work, I think it would make sense that your reaction has to be large enough that the overall net delta G, or net change of energy, is still going to be negative. Okay, so in essence, you need excess energy to go ahead and be able to do your energy unfavorable reaction and still be successful in getting products for both of your reactions. So keep that in mind. We can do a reaction coupling, and therefore we can justify or utilize energetically unfavorable reactions. Your body has to come up with this mechanism, or has to utilize this mechanism, because part of our homeostasis is not only the breakdown of products, but also the building and the maintenance of products. If you'd like, I think I took it out of the PowerPoint, so we can put it back in. Take a look at panel 3.1 in your book. It will give you lots of different examples of biological reactions and the amount of free energy that they're able to generate. There are also some mathematical problems that you can practice on to kind of see from a numerical standpoint if a reaction is going to be more negative or positive. I was not planning on doing any mathematical problems on our exam. I can if you'd like, so just let me know. I'll be more than happy to do so. For me, it's more important for us to understand the whole concept of what it meant when we say things like free energy and delta G, or if you're doing, like, for instance, obviously your free energy change. I want you to understand what's occurring. When am I releasing my energy? When I have to put energy into the system? And keep in mind that oftentimes these are linked. And speaking of the linked process, enzymes are an essential tool to be able to link an energy unfavorable reaction to an energy favorable reaction, because what the enzyme is going to do is it's going to allow us, by lowering the activation energy, to get away with putting less energy in the input of our energy unfavorable reaction, meaning that we once again are able to generate products in both of the reactions that are coupled together. Another thing that we want to take a look at is that when you take a look at your delta G, we're going to notice that the relationship between the substrate and the product will often change as the reaction proceeds, because usually what we see happening is that as the reaction occurs for a longer period of time, the concentration of substrate starts to decrease, and then obviously the amount of product that you're able to generate will start increasing. You know, that just kind of makes sense. You're using up what you're starting with and you're building up more and more product. Eventually what we see happening is that we're going to go ahead and we're going to reach equilibrium. And when you reach equilibrium, that means that your delta G is going to equal 0. And knowing your equilibrium is going to be very important, because it's going to allow us to, in essence, detect what way the reaction is going to proceed in an energy favorable or unfavorable. But it will also be able to see how far it can go, meaning how much substrate do you need to generate a certain set of products, and vice versa. So eventually what we see happening is everything will come into equilibrium when you take a look at the relationship between your substrate and your product. We also see that the standard free energy change, this is a new one, makes it possible to compare the energetics of different reactions. Now, the standard free energy change is independent of the concentration, meaning it doesn't really care how much substrate or products you have. It is going to depend on what we like to call intrinsic characteristics of all the molecules, and the molecules do have to have an ideal condition, and that ideal condition is that you're really taking a look at how the intrinsic characteristics behave when that molecule is set at 1 mole per liter. So this is where you have a certain set of molarity that comes into play. Your delta G can be calculated from your standard free energy, but that is only if you know your concentration of your reactant or your substrate. as well as your product before you go in and complete the reaction. So we do also see that there tends to be a proportional relationship between the equilibrium constant and the standard free energy symbol. My apologies. Right over here it says the equilibrium constant is directly proportionate to the free energy, the standard free energy. So here's a little table for it. And as you can see, what they've done is they've calculated and set standards between the equilibrium and the standard free energy change. And what we have over here is that oftentimes we can explain the fact that they are proportional because at equilibrium, the rate of forward movement, so building, is the same or is balanced as the rate of reversal, meaning that you're going back from product to substrate, so you're back to destroying. And then if you see that the rate is balanced between the forward and the reverse, that also means that your delta G, your free energy change, is zero. So there really isn't a net change in energy to drive the equation either way. And knowing this will then allow us to establish an equilibrium constant where, obviously, as I was mentioning to before, the ratio of the substrate is equal to that of the product. And once you're able to see that, then we can go ahead and take a look at how the individual reactants that are involved with the reaction, how their intrinsic behavior is then determined by the 1 molarity per liter. And that then allows for us to see that there is a way to predict the equilibrium constant based on what our standard free energy change is going to be all about, because they are completely related to each other. Now, also keep in mind that oftentimes, or I should say before we were kind of chit-chatting about substrate and product, we were really just kind of talking about one substrate producing one product. This often is not the case in our cells. Oftentimes the cells will have 2 if not more substrates that have to combine together to produce an overall product. So in the little example right here in figure 319, we see that our substrates of A and B have to combine together to form the product AB when they're doing their association. And when they're doing the dissociation, then of course you're breaking down your AB into the individuality of A and B. Now keep in mind that when you're doing your chemical reaction, or when you look at an example such as this, what you're trying to figure out is what the likelihood is of these molecules randomly generating enough energy that they combine together or they separate, because it's all about this kinetic energy, this energy of motion. And we've already discussed the fact that oftentimes this will not naturally occur because it has that activation energy, and that of course is where your enzymes will come into play. But that right here is a little bit above the point. What we see happening is that regardless if you have one substrate producing one product, or you have multiple substrates that combine to produce the product, as in my cases over here, so we have A and B, not just A, not just B. What we see happening is that we simply are going to have to adjust, because our collision opportunity of forming A and B is basically equal to the encounter, figuring out the rate of encounter between A times B. So right over here, I can take a look at my association and my dissociation rate, and notice how I'm multiplying my concentration of A and B. And when I do that, I'm accounting for the fact that there are 2 substrates that I have that I'm interacting with, not just one. So I'm accounting for all of my reactants and all of my products. And if I do that, which is more commonly inside of the cell, if I do that and I use that in my calculation, I see that I can also come up and come with my equilibrium constant, because there will come a time in this reaction where the concentration of the substrates is proportionate to the concentration of the products. And once you have your equilibrium constant, then it doesn't matter how many substrates you have. You can go ahead and apply the knowledge of your standard free energy change and its proportion to the equilibrium constant. You can go ahead and apply it the same way you would if you only had one reactant that produced one product. The other thing that the equilibrium constant will take a look at is it will give you a nice idea about the strength of non-covalent binding interactions. So it doesn't only give you an idea of how many bonds are being formed and broken between the substrate and the product, it also indicates the strength, or the binding strength, I should say, between the individual substrates that are interacting together. And if you take a look at the figure right here, figure 320, it gives us a little example where it gives you an analogy of 1,000 molecules of A and 1,000 molecules of B in the cytoplasm. And what we see happening here is that just by making a small change in the number of weak bonds, we will have a drastic change in how they're able to interact with each other. And that is because your binding strength will indicate the specific interaction, how many specific interactions you have between your molecules. So we can utilize that concept as well when we calculate our equilibrium constant. For sequential reactions, which is another thing that we see happening quite often in the cell, where one reaction produces a product that leads to the start of a secondary reaction. Well, if that happens, we see that the changes in free energy are what we like to call additives. So that means that they'll basically build one on top of each other, and we can still use the point of taking an energy unfavorable reaction and utilize the free energy generated by an energy favorable reaction to keep it moving. So I kind of like how your book explains it, and it just basically says an energetically unfavorable reaction can be driven by an energetically favoring, and if we see that happening, we have what we call a chemical siphon. How do you love that description? So you're basically utilizing the energy favorable reaction for its release of free energy, and then we can go ahead and use that to keep the catalase or the next step in our reaction occurring, whether that is an energy unfavorable or favorable reaction. And that's basically what we were talking about at the beginning of the chapter, is that your body, or I should say your cells, love to couple these chemical reactions together where one is favorable and the other one is unfavorable, because you can immediately transition the energy from one chemical to the next reaction. Oh, so this is a good one. So it says energy catalyzed reactions depend on rapid molecular collisions. Yes. So the question is, if we look inside a cell, we know that it's filled with cytosol. And we also know that it is just full of different molecules. So it's a very populated area to look at. And I'm thinking about that beautiful picture we saw with the different molecules, kind of look like an abstract drawing in one of our previous chapters. So we know that the cytosol is just filled with all these molecules. So how do we assure that the substrate and its proper enzymes are able to meet up? Right? How do we do that? Well, it turns out that we can rely on that kinetic energy, that energy of random motion that all molecules possess. And what we see happening is, as the cytosol is so full of all these molecules will innately start bouncing off each other. And as they're bouncing off each other, we see that they start creating these random patterns of movement. So here my little green line, I don't know why I'm writing over it with my red, but here my little green line is representing the way or the distance that the little molecule is traveling. So as you can see, it's like pinging back and forth. And as it's doing it, it's easily able to connect with all the different molecules that it's being exposed to in this densely packed area. And this is one of the ways why even if you have just a minute amount of enzyme or a minute amount of substrate available in the cell, you'll still have a pretty good chance that they're able to meet up with each other, because the bouncing from one molecule to the next generates so much movement and is able to cover a lot of distance in a short period of time, thereby increasing the likelihood that they're able to collide together and meet up together. We also see, and this is what the second statement says about, is that the non-covalent interactions allow enzymes to bind to specific molecules. And the non-covalent interactions are very important to point out, because it turns out that if you have a very strong bond between the substrate and the enzyme, chances are that the reaction will proceed from start to finish. On the other hand, if you have an unmatched substrate with the active site, we see that it's a very weak bond, allowing the substrate to easily fall off the active site, which makes sense because it's not properly fitted to it, and thereby slowing down the reaction. So here on the little illustration, this is figure 3-24, it says that enzymes cannot change the equilibrium point, so that will always stay the same, but what the enzymes will do is that they'll catalyze it in the sense that the reaction will occur at a much faster rate. Notice how large and thick this red arrow is right here, and this correlates with an enzyme-driven reaction. And because it has the ability to lower the activation energy, it can thereby speed up the overall reaction time, thereby allowing you to reach equilibrium faster, as well as produce product faster than you would in an uncatalyzed reaction. So both of these, with or without the enzyme, would reach equilibrium at some point. It's just that the enzyme catalyzed driven one will reach it a lot faster. I believe this is the end of the free energy and catalysm. As always, if you have any questions, definitely let me know. For our next part, we're going to take a look at activated carriers. So we're going to talk a little bit about some ATP molecules. All right, so I will hopefully see you, or you'll hear me in a bit for part number 3.